The smooth muscle cells, they are different than skeletal muscles or the cardiac muscles because they are smaller cells, 15 to 200 micrometer long, and their width is 5 to 10 millimeter, and they are not cylindrical, they are spindle shaped, they are flat cells, and they have single nucleus in each cell, and they have few actin and myosin, and actin and myosin, they are not in the form of sarcomere, they are arranged in bundle form, and there is no striation because they are not in linear fashion. In the skeletal muscles, the sarcomeres, they are in linear fashion, they overlap on each other, so actin overlap on actin of the other myofibrils, and the sarcomeres and myosin of one sarcomere overlap the myosin of other myofibrils. So when we see under microscope, it appears to be striated. But in case of smooth muscle, because they are arranged in different direction, they are not in linear fashion, they are not overlapping myosin and myosin, they are not overlapping, and the actin and actin, they are not overlapping, so they are not making striation in a muscle cell when we see under microscope. So actin, they are not attached to Z disc, instead they are bound with the dense bodies. The dense bodies are the proteins that are connecting actin to the intermediate filament of cytoskeleton. Cytoskeleton of the cell, we know, is made up of intermediate filament, microfilament, and the microtubule. So the intermediate filaments, they have special type of protein that is called as dense bodies. They are connecting these blue colored actin, they are attaching these blue color actin, and so these dense bodies, they are acting as Z disc, they are counterpart or replacement of Z disc. Mm, in case of smooth muscles, the sarcoplasmic reticulums are not well developed. It means there is the calcium need to come from elsewhere because sarcoplasmic reticulum are not present or not well developed to store calcium, and the T tubules are also not present in the smooth muscle cell, and we know the T tubules, they are invagination of the plasma membrane or sarcolemma, but in this case there are few ridges inside, and these ridges are like T tubule but not well developed, so they are called as cavuli, and their function is still unknown. Then calcium has to come from extracellular material. In case of skeletal muscle, the calcium was coming from sarcoplasmic reticulum within the cell. And in this case, because calcium has to come in smooth muscle, calcium has to come from extracellular material, so they show slow contraction because entry of calcium and then go to bind with the troponin to make cross bridges takes time, so contractions are always slow in smooth muscles. There is no troponin, so the active sites on G-actin are always open, but they need certain processes because myosin is different in smooth muscle. We know in skeletal muscle the myosin heavy chains, their heads had a light chain for ATP hydrolysis, but in this case, in smooth muscles, the myosin does not have the ATPase activity, so it requires other proteins for the hydrolysis of the ATPs, that is myosin, that is through myosin kinase, so it is a slow process, so the contraction of smooth muscle is slow. And then the myosin phosphatases are required in order to detach ADP from the myosin in order to break the cross bridges. That is again a slow process, so this tension is sustained. Then relaxation of smooth muscle require calcium to pump out of the cell, not into sarcoplasmic reticulum. That was in the case of skeletal muscle. So pumping out will take more time as compared to pumping in sarcoplasmic reticulum. So the muscle contractions are slow. So we see what is the difference, how the muscles they contract. If we see here a smooth muscle cell, and we can see calcium channels on smooth muscle cell membrane, then there is action potential. It means muscle is stimulated, the membrane will depolarize, there is production of action potential, and due to this depolarization the voltage gated channel, calcium channel, will get open. Calcium goes into the cell, and when it goes into cell, because there is no troponin, instead there is a protein that is called as calmodulin. So this calcium binds with calmodulin, and there is formation of a complex that is called as calcium calmodulin complex. Actually calmodulin is a protein that needs calcium as a cofactor. So this complex, that is calcium calmodulin, is actually active calmodulin. This calmodulin can activate another protein that is called as myosin light chain kinase. Myosin light chain kinase mean this protein can transfer phosphate group on myosin. So this is transferring a phosphate group on myosin. So it is activated by calcium calmodulin, because myosin itself is not having ATPase activity in smooth muscle cell, so it needs myosin kinase, that is myosin light chain kinase. So it attaches a phosphate group. Taken kinases always take a phosphate group from one molecule and transfer it to another molecule. So this myosin kinase, it is transferring phosphate group from ATP to myosin to make it energized. So then myosin, because active, there is no troponin, actin sites are always open. So when myosin is energized, it is attached with phosphate, it will get attached to actin to make cross bridges, and then there will be contraction. After that contraction, the myosin cannot release the phosphate group, so there is always need of another protein that is called as myosin light chain phosphatases. Phosphatases are the enzymes that detach phosphate group from any molecule. So myosin is a molecule, phosphate group was attached with that, so after contraction it is no more needed. So the myosin light chain phosphatases, they come and they detach this phosphate group, so the cross bridges are broken, myosin get detached from actin, and again if there is action potential, this myosin light chain kinase can again energize it. So this is the whole contraction of the smooth muscle cell. The smooth muscles can be unitary smooth muscles, or they can be multi-unit smooth muscle. Unitary means a single unit smooth muscle, or the muscle cells that are connected to each other through gap junction, or the bridges, so that if one cell is stimulated, it can pass material to, that is the action potential or the ions, to other cell to stimulate that cell, and so on. So these type of single unit or smooth muscle type of muscles, they need only one neuron that is reaching to one cell, and then if one cell is stimulated, it can pass sodium to other cells to stimulate them. That is, these single unit smooth muscle, they work in a uh, group like fashion, they can contract simultaneously in a group form. So these activities that require less precision, like the passage of food in intestine, in esophagus, in the large intestine requires a raw activity that is pushing food, so it is not that precise. So these muscles in intestine, they are working in a single unit smooth muscle cell fashion because these cells, they are interconnected. One cell is stimulated, it can pass material to another cell to stimulate it. But in case of precise movements or precise activities, they require multi-unit smooth muscle cells. For example, for focusing eyes require very specific activities in order to focus, and these muscle cells in eyes that are for focusing length or independent cell, they are not connected to each other, so neurons have to reach each and every cell to stimulate it for a precise function for its contraction. So only one cell contracts at one time in order to perform very precise function. The smooth muscles, they are under nervous control, that is the unconscious, or we can say non-conscious type of control, that is from medulla oblongata. Nerves are coming onto the smooth muscle. We know the smooth muscle, they are lining the tubes of the body, that is digestive system, reproductive system, respiratory system, circulatory system. Their tubes are lined by the smooth muscle in two layers, that is circular muscle that are controlling diameter of these tubes, and longitudinal muscle that are controlling length of these muscle or passage of material in these tubes. So the nervous control is from medulla oblongata because it is for unconscious activity or non-conscious activity. So the nerves, they are coming and they are controlling the smooth muscle. They are also under the control of hormones. For example, oxytocin, epinephrine, norepinephrine, they can increase diameter of the blood vessels. They oxytocin like work in uh, the during birth and for the ejaculation and for the secretion of material from glands. So these hormones, they can turn those muscles, smooth muscles, and like epinephrine, norepinephrine, oxytocin, antidiuretic hormone, they are for the that is also called as vasopressin. They can increase or decrease diameter of these tubes or the length of these tubes. The smooth muscles have four properties. They may autorhythmic. Few of the smooth muscle, they can produce auto activity, mean they can contract themselves, but not all smooth muscle. And smooth muscle, when they are stretched, suddenly they will contract back. They have a continuous tone like skeletal muscle. Few of their cross bridges between actin and myosin, they are always formed in order to keep the tubes open, a little stretch on the tubes so that the tubes remain open for the flow of material. They may apply constant force to the when a hormone, as we said, the oxytocin or epinephrine, they may bind with the smooth muscle cell, and they bind to their specific receptor. They are metabotropic receptors or channels. So the hormone bind with metabotropic channels, and then they are G protein coupled receptors. So they are then linked with phospholipase C. So G protein alpha unit, as we know previously, alpha unit goes to phospholipase, and phospholipase can break down phospholipid, that is phosphatidylinositol bisphosphate into inositol triphosphate and diacylglycerol. So diacylglycerol is not visible here. It is inositol triphosphate. So inositol triphosphate can open calcium channel in the endoplasmic reticulum. So few of the calcium they may go into the uh, cell from endoplasmic reticulum, and IP3 also open the calcium channel in membrane, so the calcium can come into the membrane for into the cell for activation of calmodulin and then subsequently contraction. Same is done by some hormone. Some hormones they go to bind with receptor that are coupled with G protein coupled receptor, and G protein alpha unit of G protein may go directly to calcium channels to open the calcium channel and to let the calcium come into the cell to bind with calmodulin. Same if there is change in action potential, then calcium voltage channels may open and the calcium comes into the cell to activate the calmodulin. So these are three modes of entry of calcium into the smooth muscle, and it is the activation of smooth muscle.